Welcome friends to a new session on molecular biology. In this session, we will discuss the initiation, elongation and termination of transcription in eukaryotes. In prokaryotes and eukaryotes, the process of RNA synthesis is similar, but the process is considerably more complex in eukaryotes. In eukaryotes, RNA is synthesized in nucleus and must be transported to the cytoplasm for translation. In prokaryotes, mRNAs often contain the coding regions of two or more genes. Such mRNAs are said to be multigenic, that is, controlled by multiple genes. On the other hand, many of the eukaryotic transcripts that contain the coding region of a single gene are monogenic. In eukaryotes, the bulk of primary transcripts in a nucleus are called heterogeneous nuclear RNA, that is hnRNA, because of the considerable variation in the sizes of RNA molecules present. Majority of these hnRNAs are non-coding intron sequences which are excised from the primary transcripts and undergoes various processes before leaving the nucleus. The RNA transcripts are coated with RNA binding proteins during or immediately after their synthesis in eukaryotes. These proteins protect the gene transcripts from degradation by ribonucleases, that is, enzymes that degrade RNA molecules. The average half-life of E. coli is less than 5 minutes. The average half-life of a gene transcript in eukaryotes is about 5 hours. The RNA molecules that are translated on ribosomes are called messenger RNAs, that is mRNAs. During transcription, one strand of gene is used as a template to synthesize a complementary strand of RNA is called the gene transcript. In prokaryotes, the primary transcript is equivalent to the mRNA molecule. In eukaryotes, the primary transcripts must be processed by the excision of specific sequences and the modification of both termini before they can be translated. General features of RNA synthesis. RNA synthesis occurs by a mechanism that is very similar to that of DNA synthesis, except that 1. The precursors are ribonucleoside triphosphate rather than deoxyribonucleoside triphosphates. 2. Only one strand of DNA is used as a template for the synthesis of a complementary RNA chain in any given region. And 3. RNA chain can be initiated de novo without any requirement for a primer strand. The RNA molecule produced will be complementary and identical to DNA template strand, except that uridine residues replace thymidines to the DNA non-template strand. If the RNA molecule is an mRNA, it will specify amino acids in the protein gene product. This specification is accomplished by nucleotide triplets called codons. Each codon specifies a single amino acid in a polypeptide chain. Therefore, mRNA molecules are coding strands of RNA. They are also called sense strands of RNA because their nucleotide sequence makes sense in that they specify sequences of amino acids in the protein gene products. An RNA molecule that is complementary to mRNA is referred to as antisense RNA. The synthesis of RNA chains like DNA chains is in the 5 prime to 3 prime direction with the addition of ribonucleotides to the 3 prime OH group at the end of the chain. The reaction involves a nucleophilic attack by the 3 prime OH group on the nucleotidal phosphorus atom of the ribonucleoside triphosphate precursor with the elimination of pyrophosphate as in DNA synthesis. This reaction is catalyzed by the enzyme RNA polymerases. The overall reaction is as follows. Here N is the number of moles of ribonucleotides triphosphate RTP consumed. Ribonucleotides monophosphate RIMP incorporated into RNA and pyrophosphate produced. RNA polymerases initiate transcription at specific nucleotide sequences called promoters, which are different in prokaryotes and eukaryotes. RNA synthesis takes place within a locally unwound segment of DNA, sometimes called transcription bubble, which is produced by RNA polymerase.